Hi, my name is QinQin Yu, and I'm a graduate student in the Halichek Lab at UC Berkeley. Today I'll be talking to you about the mutability of demographic noise in microbial colonies. Demographic noise is the stochasticity due to random births and deaths in a population. It's sometimes also known as genetic drift, neutral drift, or just drift. And we're interested in studying demographic noise because it can compete with other evolutionary forces, such as selection, mutation, and recombination. In particular, some consequences of demographic noise are that it can weaken the efficacy of natural selection, reduce neutral genetic diversity, and promote the evolution and maintenance of cooperation. Experimentally, systems where demographic noise has been shown to influence evolution include in the transmission of viruses, in stem cells and intestinal crypts, and in the transmission of human mitochondrial DNA from mother to child. Now, these are all examples that involve small population sizes at some point in time. And you might be wondering, is demographic noise at all important in larger population sizes, which so often is the case for microbial populations? It turns out that the answer is yes for some types of populations. One example is range expansions. Range expansions are spatially structured populations that expand out from some central location. And intuitively, we can understand why demographic noise can be enhanced in these systems by imagining a simple one-dimensional range expansion. An individual in the bulk of the range will not have sufficient space to place its offspring, whereas one at the front would. And this reduces the effective population size, or the population size that can produce offspring for the next generation. This in turn increases the strength of demographic noise. Microbial range expansions are also interesting to study because they're prevalent in the natural world, such as in the form of natural microbial mats and in biofilm infections, which pose a major medical problem. Now, what sets the strength of demographic noise? I've told you that conventionally, it's thought to be set by the population size, and also by environmental conditions, which can alter the population size. Recent work has shown that, in addition, in microbial colonies, demographic noise can be influenced by genetics. And in this experiment, they've compared two different strains of E. coli, and each strain has been transformed by two neutral fluorescent labels. And initially, the number of sectors in the two strain backgrounds is similar, but due to stochasticity, the sectors annihilate from the colony front. And higher stochasticity due to higher demographic noise leads to more loss of sectors. So in this case, in MG 1655, there's many fewer sectors, which indicates a higher strength of demographic noise. Additionally, the differences in demographic noise seen between these two different strains has been shown to influence the efficacy of selection by changing the establishment probability on the different strain backgrounds. So because we see that genetics can change demographic noise, it leads us to ask the question, can demographic noise evolve? Can it change due to mutations? And in order to answer this question, we have to answer a more specific question, which is how much can single mutational steps change the strength of demographic noise in microbial colonies? Our goal will be to measure the strength of demographic noise in a large number of single gene deletion strains from the E. coli Kyo collection. And in order to do this, we developed a label-free method for measuring demographic noise in colonies. We use fluorescent beads that are about the same size of as the cells, and as the colony grows, it pushes the beads, which track a subset of cellular lineages. We can zoom in at the single cell level to check that the beads are closely following the neighboring cell trajectories. And here, the bead is tracked in black and neighboring cell trajectories are tracked in colors. And what we see over the course of this 1-hour movie is that the bead indeed closely tracks neighboring cell trajectories. We can then do single particle tracking of these beads, and then we can quantify the amount of wandering by measuring the mean squared displacement, or the displacement of the trajectory from an average direction of motion. We then validated that we can indeed measure the strength of demographic noise using this method by comparing to the existing method of mixing 2 neutral fluorescently labeled strains. So we did this by transforming each strain background with a plasmid that contained either YFP or CFP, mixing them together at equal proportions and growing colonies. We then defined the fraction of diversity preserved as the number of sectors normalized by the initial number of cells at the inoculum. So when we plot these 2 quantities against one another, we would expect that higher mean squared displacement leads to a lower fraction of diversity preserved. And indeed, this is what we see for a subset of 10 strains. So this shows us that our method is able to capture differences in demographic noise between different strains of E. coli. Next, we wanted to characterize the distribution of strengths of demographic noise due to single gene deletion strains. And we did this by randomly selecting a subset of 200 strains from the E. coli Kyo collection. And when we measured the strength of demographic noise in these strains, we saw a significantly different distribution than that of the wild type, which characterizes the amount of experimental noise in our system. Furthermore, we saw that the median of the single gene deletion distribution was lower than that of the wild type. And this suggests that a typical single gene deletion mutant will tend to decrease the strength of demographic noise. So next we wanted to know whether this distribution of strengths of demographic noise was wide enough to affect adaptation. And we did this by looking at the establishment probability of a beneficial mutation on the different single gene deletion backgrounds. And theory predicts that demographic noise should weaken the establishment probability. The theory prediction in colonies is shown here, and while it looks quite complicated, it depends on just 3 parameters: the diffusion coefficient from single cell trajectory wandering, the fitness coefficient of the beneficial mutant, and the initial fraction of the beneficial mutant in the population. To gain intuition for this prediction, we can plot the establishment probability as a function of the fitness of the beneficial mutant on the background strain. And we see, as expected, that higher fitness mutants tend to have a higher probability of establishing in the population. We can also look across different diffusion coefficients for the same fitness coefficient, and we see that higher diffusion coefficients decrease the establishment probability. And this shows that demographic noise weakens the establishment probability of beneficial mutants. So to test this prediction for single gene deletion strains, we took single gene deletion backgrounds and transformed them either with a plasmid that contained YFP and a resistance gene for the antibiotic chloramphenicol, or just a CFP gene. And then we mixed the resistant type at a low proportion with the sensitive type and grew them as colonies in the presence of different concentrations of chloramphenicol to modulate the selection coefficient of the beneficial mutant. We then measured the establishment probability as the number of resistant sectors normalized by the initial number of resistant mutants. And here's an example for the strain background of the E. coli Kyo collection. And we see a different number of sectors establishing when the beneficial resistant mutant has a different fitness coefficient. And here's an example on a different strain background, where we see many more beneficial sectors establishing. So we can plot the establishment probability for a beneficial mutant on each of these strain backgrounds as a function of the mean square displacement of that strain background that we measured using beat trajectories. We did this measurement for a subset of about 10 strains, and we indeed see a negative correlation between the two quantities. So what this shows us is that single gene deletion mutants generate sufficient variation in demographic noise to affect adaptation. So in summary, I've told you about a label-free method to measure demographic noise in colonies. I've showed you that single gene deletions exhibit a distribution of strengths of demographic noise that's sufficiently different from just experimental noise. We've also seen that the variation in demographic noise can be explained by colony-level phenotypes, which I didn't have time to talk about during my presentation, but I'd be happy to talk about in the questions or in the chat. And finally, we found that single gene deletions generate sufficient variation in demographic noise to affect adaptation. And in sum, our results show that demographic noise can be an evolvable property of a population that changes due to mutations. And this challenges the conventional view that demographic noise is just a static property of the population. Finally, I'd like to acknowledge key players in this work, including my advisor, Oscar Halachek, former grad student Matthew Kralka, postdoc Marie-Cecilia Duvernoy, and undergraduate Megan Souza, as well as Arvel Harbeck, a statistician and postdoc at Columbia University, who helped with some of the statistical analyses in this work. And thank you very much for your attention. I'd be happy to take any questions.
All right, thank you much, QinQin. So we have one question in the Q of A so far from Grant. So Grant's asking, I was curious about the availability of gene knockouts that reduce displacement below the wild type. Would you ultimately expect there to be perhaps second order selection for mutations that reduce demographic noise? Or is there some reason why a higher level of demographic noise would be favored?
Yeah, so I think that is indeed what my expectation would be, that there would be selection for strains with lower demographic noise because they have a higher establishment probability for beneficial mutants that arise on that background. But I do think that the question might be more complicated in different scenarios. Like, for instance, in a clonal interference situation, I'm not totally sure what I would expect to be in what direction demographic noise I would expect to be selected upon. So I think it's definitely an open question that we're also very interested in.
Awesome. And I'm just going to follow up with one question that popped in my head. So you have the KO collection. Have you looked at any like pathways or tried to group anything in terms of larger systems to generate noise there?
Yeah, so we have studied the genotypes that were enriched for demographic noise and tried to see if there were any pathways that were enriched for. And when we did this using statistics, we actually didn't come up with any pathways that were enriched for in the extreme values of demographic noise. And so it might be possible, it might be the case that we just need to test more knockouts, and that would give us more statistical power to detect these differences. But it might also be that the phenotypes on the colony and single cell level, which I didn't have time to talk about, but seem to be well correlated with demographic noise, it might be that these phenotypes are much more determinants of demographic noise than the actual genotypic changes or metabolic changes.
Awesome, thank you. So we've got a couple more questions that I think I'm gonna post to the Rocket chat because we're slightly out of time. So thank you very much again.
Thank you.
And we're gonna move on to our next speaker,